And of course, it was used for a very, very long time until people made ibuprofen, right? So ibuprofen overtook aspirin because it had much lesser side effects, right? For example, aspirin cannot be given to kids, right? So and later in the again 1970s, 80s, again, the aspirin came about, but not as a NSAID, non-steroidal anti-inflammatory drug, but as a blood thinner, right? So until this point, right, about 1985 or late 1990s or early 1990s, right, is when we knew until 1800s, right, we did not know what is responsible, right? For 100 years from then, we knew, something, one particular component is responsible, and then we started making better ones, right? So in the late 2000, the later part of the 20th century, basically we started learning about what it actually does, right? Okay, we consume a drug, right? What it actually does in the molecule, does in the body, right? So how does it treat, or how does it manage pain, or how do we cure, and so on? How does it work? We started understanding with the advent of the molecular biology technique. We started understanding about the target molecules in cells, and then where it binds, and then once it binds, is it possible to use that information also and come up with much better drugs, and so on, right? So this is in some sense not just the story of aspirin, but the story of NSAID over years, where initially we did not know anything, and then we knew the molecular structure of the aspirin or the small molecule, and then the information of the biology involved, or the biological pathways that are happening in the body, and so on, right? So all these actually revolutionized drug discovery. These are different phases in drug discovery and development where all these techniques was used. But then one can ask, okay, so initially we did not know anything, right? And then we came about this chemical revolution, and then the biotechnological revolution, and then now we are talking about the evolution of AI. But then why does why the drugs still do not come very fast or very, very early in the market, right? Why do they, and so on. So we'll talk about it briefly. Okay, so I'm actually taking a lot of time already, so I'm going to skip few of these. But the idea is this, right? This is what I was trying to tell, that the whole point of the discussion is this. Initially, everything was empirical, and then second is the understanding of the drug, and then understanding of the drug and the target, and then the relevance of that target in the disease, and so on, right? And of course, different kinds of industry came about, and then started participating in this. Of course, when we talk about experiments, or when we talk about what we actually do to come up with a drug, these are different kinds of techniques that are involved. But at the larger level, we did all the tests only at the organism level, and then started doing at tissue level, cellular level, molecular level, and so on. So now, why is it more complicated? We are not only there. I mean, in principle, it's not that we do only the molecular level, but we still do the whole thing, right? And then again, it's just not that. Okay, so what is the drug discovery process as we understand today? This is a slightly complicated diagram, but I will go to the next one and simplify it. Let's say I have a disease, new disease or existing disease or whatever, and what do we want to do? What we want to do is to come up with a drug molecule that is marketable. So that means it has to pass through several layers of safety, regulatory approvals, and all kinds of things, right? So before you can get there, a lot of science needs to be done. So all this process, is people contribute, right? People contribute to different stages in this process. Again, it's a very simplistic representation, right? Typically what they say is it takes about 10 years. It takes $1 billion or so for coming up with a single molecule with a lot of failures in between. Okay, so I'll tell you what again, what is the criteria? The criteria is basically this. Typically, at least in the initially, where what we want to worry about is for a drug to actually act, for it to actually work. All of these things should work. For example, if it's if the drug needs to be administered along the oral route, So basically, it has to go from your, we just pop up the pill and then forget about it, right? So it has to go from the digestive system to the circulatory system. Once it goes into the circulatory system, it has to go through the first pass of the liver, and then, it should not, form complexes with the proteins present in the blood, right? So, and then the heart should be able to, distribute this around this. And once the drug action has happened, it has to be metabolized and then excreted. Again, this is not true for all the drugs that are there, but most of them. Okay, so what is the role of computations? The computations is where, I have to stand here, right? Okay. So the computations is where you have this target, which is given in all those blue, red, and gray colors. And then now we want to come up with molecules which will go and sit there for a reasonable amount of time, because of which there is a pharmacological activity. Okay.